Hello, in this video we will discuss about the Illumina sequence of DNA. And what is this? This is the next generation sequence after a sequencing after the Sanger method of sequence. If you want that Sanger method of sequence video, the link in description below. So this is the cell. Let's begin to understand the sample cell. This is sample cell contain nucleus, cytoplasm, cell membrane, and chromosome. Chromosome contain DNA, and DNA is made up of adenine, thymine, guanine, and cytosine nitrogenous bases, which that adenine always bind with a thymine, antiparallel 5' to 3' and antiparallel 3' to 5'. So let's begin to understand the step of Illumina sequencing. Number one is the sample preparation. Number two is the cluster generation. Third is the sequencing. And the fourth one is the analysis of data. Analysis of data is due to the in this way, which that is totally different as compared to the Sanger method of DNA sequence. So there are a number of different for preparation of samples. So number one is the sample preparation, how it's possible. So after the DNA extraction, the fragmentation will lead to add the adapter. Adapter add the P1 and P2. Remember, this adapter will bind with the end of the DNA 3' and 5'. And here I am taking the adenine, guanine, cytosine, thymine sequence, which that should be amplified, and the specific primer should be required for this amplification. So remember, an other segment will not amplify because the specific primer will not bind. So the additional motifs introduces such as sequencing binding site indicates and regional complementary to the flow cell oligos. So the flow cell oligos is the cluster formation we will discuss also. So in this way here is the binding of the additional motifs introduced in this manner. And after this is the complete segment preparation, and there a lot of segment will be bound by like this, but the specific segment will be amplified because we are searching the specific gene segment. So let's cluster formation we will discuss. So the clustering is a process of where each fragment of molecule is isothermally amplified. The flow cell is a glass slide with lens. This lens, each lens is a channel coated with a LAN and composed of two type of oligos. Two type of oligos is oligo 1 and oligo 2. So in this way we will discuss the flow cell. This is the flow cell is a glass slide with lens. Each lens is a channel coated with a LAN composed of two type of oligos. Let's begin to understand what is the oligos. Oligos is basically is the adapter like. Basically adapter will add the oligos, which that flow cell containing character is the oligo. The two type of oligo we will introduce like a adapter P1 and P2. So in this way this is the prepared the flow cell, which that is composed of O1 and O2 oligos. So in this way the basically is the DNA, which that is required DNA. For example, there a lot of DNA will bind, but the primer specific DNA amplification will occur. So in this way here is you can see this the P2 and P1 will ultimately bind because the oligos will bind easily. After this binding, there are several number of oligos are present, two types, which that polymerase enzyme used to amplify. So the polymerization after the binding of the primer, the specific primer, because a lot of segments are present, but the specific primer we require for the amplification of the specific segment. So this is the polymerization. After the polymerization, the original strand is here, and while this is the new strand synthesis due to the polymerization through polymerase enzyme. So remember. And after this, the denaturation will occur. The original strand will be denatured, and washed away, while the new strand will maintain. After the new strand will maintain, which that will be oligos will bind, remember, with the O2 with O2, which that are present already in the flow cell. So the flow cell So the flow cell contain basically is the two type of oligos. So here is the washed away the original strand but the denaturation after. So the new strand will maintain and bind with the surface of the flow cell. After the repeating process, amplification will occur, the clonal amplification of all fragments, which that is equal to the new strand and bind with the oligo 1 or 2. So in this way, the process we will learn. So hybridization will occur. So the hybridization mean basically the bridge formation will occur. In this way, the oligo 2 will bond with the oligo 2. So remember, after this a polymerase enzyme with a specific primer will bind to a synthesis of new strand, which that toward the oligo 1 site. And in this way the bridge formation will occur, and in this way the denaturation will again occur. After this denaturation, the two single strand will be separate, and after the separation of the two single strand, the repeating process amplification will occur again and again in the cycle, and the clonal amplification of all fragments will occur. But the sum will bind with the oligo 1 in the surface of the flow cell, while the other will bind with the oligo 1, oligo 2. So here you can see the repeating process amplification. The repeating process will occur. In this way, the repeat again and again this process, the bridge formation and hybridization will lead to clonal amplification of all fragments, and basically all fragments mean required fragment due to the primer specific fragment will be amplified. So after this, the after bridge amplification, the reverse strands are cleaved and washed away. What that means? That means the sum which that bond with the oligo 2 and some will bond with the oligo 1 in the surface of the cell, and in this way the basically is the after bridge formation, after bridge amplification, the reverse strands are cleaved and washed away, washed off, sorry, washed off, not washed away. But remember, this is the leaving only the forward strand, but the reverse strand will cleaved and washed away, for washed off. So it will be delete from the flow cell, while the leaving only the forward strands, which that contain 3 prime end block to preserve the unwanted primering, and in this way the unwanted primering will not bind, and in this way the specific amplification will occur. So after this is the leaving. After, so let's begin to understand the sequencing method and sequencing process we will learn. So again I am repeating, the after bridge amplification, the reverse strand are cleaved and washed off, while the leaving only the forward strand 3 prime and block to preserve the unwanted primary binding. So the sequencing is due to the production of color with the each nucleotide binding to produce the color. So remember, this is the adenine, guanine, cytosine and thymine, which does contain adenine orange color, guanine is blue, pink and thymine contain green. So in this way, the fluorophore containing nucleotide for indicate to sequence of each fragment. So when we'll bind with the front of the adenine, the thymine can be bind by this strand. So remember, when thymine will bind, will show the green color in fluorescence. Sorry. So bioluminescence will produce this fluorophore, which that detect in the data analytics. So in this way, again and again, this binding to show the color. So remember, and cycle determine the length of the read. The length of the read mean each nucleotide will bind to show the color, so we can detect that nucleotide will bind 1, 2, 3, 4 and show the color, and the type of nucleotide adenine, thymine, guanine, cytosine, which is attaching one by one, and the sequence we can determine easily. So the cycle determines the length of the read, the emission wavelength along with the signal intensity determine the base call. So the base call determination, for example, one base add, for example, thymine will produce green color. So we can say in this time the thymine will bind, and if the cytosine will bind to produce blue color after the thymine, thus in this way we can say that cytosine. So remember, this is the basically not original strand. So the original strand copies of the original strand will be annealing and extension. So after uh, all identical strand read simultaneously, and hundreds of millions of clusters are sequenced in a massively parallel process, and after the completion of first read, the read product washed away, and the read product wash away. In this step, index 1 read primer is introduced and hybridized to the template. The read is generated similar. So in this way, here is the washed away, and the sequencing by synthesis here is, you can see in this diagram, and after this again the washed away the new strand, but remember the primer will again bind to synthesize new strand again to color, and the concentration of the production and the result we can understand more. So in this way, again and again the repeating process, but remember that washed away the new strand. But after this washed away the new strand, the amplification of the DNA will more. And in this way you can understand again, this is the oligo 2 and oligo 1, but the oligo 2 will bind toward the oligo 2, because oligo 2, oligo 2, oligo 2, and in this way the washed away the basically is the polymerase enzyme number 1 will synthesize the new strand, and the original strand washed away, and only original and protect 3 prime again. And in this way, remember, this is the process which that continuously cycle, and after bridge amplification the reverse strand are cleaved and washed off. So again this process will occur, which that we had discussed in the previous diagram. So after this, here you can see the analysis, you can understand here is the biological sampling, genomic DNA isolation, fragmentation, and genomic DNA. So the align overlapping sequences, which that is here you can understand, and generate the counting sequence, which that show this is the fragment reading, and this fragment reading is exact, for example. So in this way, after the sampling is it show that gene 1 and G2 and forward and reverse read are paired. We can pair it forward and reverse read. So in this manner, the Illumina sequencing of DNA show the exact number of the bases on the basis of sequences, and we can determine the segment with the adenine, guanine, cytosine, thymine, and we can determine that complete sequence with the number of bases, and we can identify more strongly as compared to the Sanger method of DNA sequencing. So the genomic DNA can be, and genomic data can be transferred, store, analyze, and share in base space sequence hub. So I hope you make sense about the Illumina sequencing of DNA. This is the totally different as compared to the Sanger method of DNA sequence, because Sanger method of DNA is different. So thanks for watching. Bye.